Welcome to CLS 307. I'm Dr. Nasser and I will be your instructor this semester for this course. I hope you're all well. You have had a good rest. You're back here to go and get into action again. I have a little bit house, work that I'm going to take care of right now before we get started with the syllabus and the lecture. How many of you are not registered in the course? Raise your hand, please. Okay, unfortunately, I will not be able to accommodate all of you. I have already had a waiting list that I have made. I am going to go over the names. If you are present in the class, you will get the spot. If you are not present in the class, I'm going to give you a spot to somebody else. So, is Juliana Leon is here? Okay. This is your permission number. I need you to register by the end of this week. Okay. Mm-hmm. It's Justin Catapong here. I want you to get registered by the end of this week. Okay. This is your permission number. Michael Mendez. These are set up. Okay. Thank you. Okay, this is it. People who just got their permission number, you have to register in Tuesday's laboratory. You're all going to come to Tuesday laboratory, okay? If you are not registered in the course, I am very sorry about this, but we have a cap of 46 people and we cannot take any more students. You got to realize that our laboratory only has 23 spots. We have 2 sections. And if you are not registered and you don't have a permission number, we cannot accommodate you. However, I got to point out that because of this problem, this overwhelming demand, which I understand, we have decided to go ahead and offer CLS 304 as well as CLS 307 this summer. So you do not have to wait till fall semester that's going to come up. You can go ahead and register in the courses. They're not going to be offered concomitantly. They're going to be offered first. We're going to offer one class. Then after that one is going to be finished, we're going to offer the other one. Of course, it's going to be the summer session, so it's going to be compact. But you're going to come to school a little bit more often. Right now, your lecture is once a week. You will have to have your lecture twice a week during the summer. So don't despair. But this is what it is. We have limited number of chairs, seats available in the laboratory. Otherwise, we could take the lecture, but we're not going to be able to take you in the lab, and that's not going to work out. Okay? So if you are not registered in the course, you may leave now. Okay? But do not give up. Okay? All right. For those of you who just got the permission, Michael and Juliana, you will come to. Did you miss the laboratory?
I did.
On Tuesday, and you missed the laboratory on Tuesday. What I want you to do is that both of you come to the Thursday laboratory. I'm going to go ahead and squeeze you in the corner someplace. But next week you're going to come on Tuesday laboratory, both of you. Okay? Justin, you were there until. Okay. Let's get started, folks. Let me find out where the light is in this room. Is it too dark for you? Should we go ahead and move the curtain? You okay? Don't fall asleep on me. This is not going to work out. No, Michael. The other way. The other way. Sorry. Open it up. Really good. That's all right. I like it romantic, but I don't think it's going to be a good idea. Okay, folks. as I said, I'm Dr. Nasser, and I'm looking forward to, take this journey with you guys. I have already sent everybody email, announcement that's going to be on Blackboard. So I have told you a little bit about myself. So if you want to know more or you have not seen the email, go ahead and read it. This is going to be a 4-hour unit course, CLS 307. Okay, it's going to be one of the important classes that you have to take in CLS program. Okay, and of course the time for the lecture and the corresponding laboratory is going to be up there. Make sure that you show up in your laboratory. Let me tell you before I forget, we need you to have lab coats when you come to the laboratory. Now the first week we are not going to be very strict. However, from next week when you come to the laboratory, when you're sitting here, settling down in your spots, I expect everybody to have their lab coats on. Okay? This room is quite weird, so I have to move around because I cannot see the screen well. So let's just move on. So the information is up there. There's nothing, everything is self-explanatory up there. So you would be able to go and contact me. This is my office. It's going to be in Welsh Hall. If you know Professor Jackson Harris, my office is very close toward the end of that hallway on the, if you're standing in front of Professor Harris's office, my office is going to be on the right side, okay? A330C, my phone number is there, and my email is here. Email is going to be the best way for you to contact me. Email is going to be the best way for you to contact me because I usually check my email many times a day, 7 days a week, and I will respond to you as soon as I get your email. But if you leave a phone message for me, don't expect me to respond to you immediately because I will get the message when I am in my office. If you send me, I'm not going to come here usually on Friday. If I'm here on Friday, I'm going to do research. I'm not going to go and I'm going to be more in the laboratory. So if you send me a message on Friday, don't expect me to go and respond to you until Monday or Tuesday. Okay? But if you email me, I will get back to you as soon as possible. Okay, my office hours are here. My office hours is for you people to come and visit with me. My office hours is for you people who have questions to come and ask questions. My office hours are not for sitting in front of the computer and surfing the net. My office hours is to go and help you folks. So when the end of the semester comes, and this has happened before, that's why I'm repeating this. When the students come and tell me, I didn't understand this acidity and basicity concept in the beginning of the semester. That's why I lost 10 points in my exam. That's why I'm going to be failing this course at the end of the semester. I will tell that person it is your own fault. Because I didn't see you in my office hours. Because if you come to my office hours, you have a question, I'll be damned if I not make sure you learn and understand what the problem is before you leave the office. I will make sure of that to explain it to you. So it's your responsibility. I love to see you in my office during my office hours, okay? So please come and visit me, come and talk to me. I want to know every one of you, and I will. Believe it or not, everybody doubt me, but I will get to know every one of your names within, I would say, a month down the road. I will know every one of you by name, okay? And you don't believe me, you will see that. However, I want to get to know you. I'm your teacher. I'm not an obstacle on your path towards success. I'm here to guide you. But if you don't ask for the guidance, you're wasting your money, you're wasting your time. I'm here to help you. Come and ask for help if you need one. These are going to be the textbooks. And both of these books, the Atlas as well as the textbook, they are available for free online. If you go to the Blackboard, and let me tell you this, we're going to use Blackboard very actively. If you have not logged into Blackboard, make sure you do so and check the web page because all the lectures, all the laboratories, all the supplementary material, your grades, announcements, everything is going to be there. It would be your responsibility at least 2 or 3 times a week. Just log in, see whether or not there's anything new. Because continuously I'm going to go and populate, add more lectures, add more presentations. I will actively go and update the files on the Blackboard. So make sure that you take a look at it, okay? You don't have to print the lectures if you want to. And I see that, I am not from a very wealthy family. So I know that how much it costs to go to school. You know, I paid my way through the school like many of you are doing it. So I see sometimes you folks come to the class and you have put one page, one slide of the PowerPoint per page in color. That's going to cost you a lot by the end of the semester. You either can, part of this, you either can put 3 or 4 slides per page. You don't have to have it in color. And if you don't want to go in and print it, you can do this. You can bring your laptop, you can bring your iPad, you can bring these gadgets that I don't know what they are called, and you can use it in the class with one condition. The purpose is to use it for the class. The purpose is not for you to be on the Facebook. The purpose is not for you. If that is that important for you, don't come to the class. Okay? This is a university class. This is not high school. I don't want anybody who doesn't want to be here to be here. Okay? If you can pass the exams without coming to the class, that's fine with me. I will promise you will not. Not because I'm not going to give it to you, because being in the class is going to be very critical for you to understand what is going on. Okay? As you can see, I got this ugly gadget up here. That's a microphone. And I'm spying on every one of you. Okay? No, I'm not. I am recording everything that is happening right now. Okay? So the screen is being recorded, my voice is being recorded, and as soon as I'm done, I am going to go to render this file and I'm going to put it on Blackboard. So let's say that next week you are going to have your first quiz. And your first quiz is going to be basically consist of everything that we talked about this week in the lecture and in the laboratory. Okay? You want to go ahead and check what we covered in the lecture. You just want to review the material. You're going to go ahead and click on the link. You're going to go to YouTube, and you're going to have exactly what is transpiring right now. Okay? So make sure that this is going to get uploaded. Every lecture is going to be uploaded probably within 2 days after the lecture is done. As soon as I get the file, I'm going to go ahead and put it on Blackboard. I put it there for you folks who want to review the material. Okay? I am not putting it there for people who just don't want it. It's just a sunny, beautiful day. They want to go ahead and play soccer. Believe me, I love to do that too. But the idea is that that's not the purpose of those files. If you are not going to show up in the class because you think you got the files and that's the only purpose, you are making a big mistake. Now, if somebody has an emergency, just a few minutes ago we had somebody had to leave because of an emergency, it's okay. That's the purpose too, for you to be able to not fall behind. But it would be your responsibility to catch up with the material that you miss. All right. Official course description and learning outcomes, they're up there. Bottom line is going to be very simple. You can read these, you can ask questions if you have, but the bottom line is that we are going to learn about the human body. We are going to learn about a specific part of the human body. We are going to go ahead and learn about that part of the human body and its association with the rest of the human body. We are going to learn a lot about blood. Don't underestimate the blood. There is a lot of information that is going to be covered in this lecture. You're going to understand how the blood is going to interact with other tissues. You're going to be able to understand how it's going to go ahead and interact with lymphatic tissue. You are going to understand how different components of blood are going to be able to work together in order to maintain homeostasis. You are going to learn a lot about that topic. But if you want the details, you can go ahead and read that, what I have put the learning outcomes, and you can ask any questions that you have. Next time when you come to the lecture, I'll be glad to answer that. Now, attendance, again, if you are trying to pass this class, is mandatory. If you're not planning to pass this class, it is not mandatory. Let's put it that way, okay? The grading criteria is pretty straightforward. I've indicated up there how you're going to determine your final grade. Before I forget, let me tell you, if you are, by the end of the semester, you get an A in this class, and you want a recommendation. The reason that I'm saying that, some students are coming to me at the end of the semester and they're asking for a recommendation letter. And I feel really bad because I can see that the student is tied down in a bad situation, but I am not going to write anybody who doesn't get an A a recommendation letter in my class. If you get an A, I'll be glad to do so. But if you get anything below an A, I will not do that, because last semester I wrote 13 recommendation letters for everybody who got an A in my classes. It took me 1 hour for each letter. That's going to be 13 hours that I spent, and I love to do it. I will do anything for those of you who are willing to go in and walk the walk. I will do anything for you. But again, it's going to take me a lot of time to write those letters. So if you want the recommendation letter, you got to start now. An A would guarantee you 1. Okay? I am not one of those faculties or one of those people that has a template and all they do is that they change the name and they send a recommendation letter. I write a recommendation letter for you, who you are, what's your strengths, what's your weaknesses, what's your potential. So it's very important to get an A if you want one from me. If you don't want, then that's fine. How we are going to go ahead and determine this grade? We are going to go ahead and determine this grade based on 800 points. We can see that 800 points in here. There are going to be 4 exams, lecture exams, 3 exams, lecture exams throughout the semester, and there's going to be 1 final exam. That's going to be 400 points. We are going to have a midterm laboratory and a final laboratory. Each one of them is going to be 100 points. That's going to be 200 points. We're also going to go and have lab worksheets. As you come to the lab, not the first and second lab, but from the 3rd lab on, you have a worksheet. You will take it home after the lab is done. You have to fill out the information. You bring it back. I will grade them. And it's an easy, this is an easy point that as long as you do your homework, you get your work done, you would be able to go ahead and get all those points. Okay? If you don't get it right, then you're going to miss those points. And finally, and in my opinion, the most important one is going to be 100 points for weekly quizzes. You will receive a quiz every week. I will give your quizzes in your laboratory. The quiz will be administered in the beginning of each lab. Okay? I'm not gonna, we don't have time for to give quizzes everybody in the lecture. I'm gonna do it in the laboratory. So Tuesday people, you're gonna go ahead and get your quiz next Tuesday. Thursday people, you're gonna get your quiz next Thursday. Okay? Question?
No, I just wanted to ask.
Okay.
Sorry.
So, why am I saying it's the most important part? Let me explain to you. Okay? What is going to be covered in each quiz? Next week, what is going to be covered in your quiz? You want to know that, right? Everything that we have talked about this week in the lecture and laboratory. What is going to be in your quiz 2 weeks down the road? Everything that we covered in the lecture and laboratory next week. Am I going to go in and ask you a question about this week, what I'm doing right now, 2 weeks down the road? Not in your quizzes. It is very unlikely I give you a question that's not going to cover the material from the week before. Now, what a smart person, in my opinion, would do is this. If you prepare yourself for the material each week, think about it, guys. Think about it, because if it doesn't make sense, it doesn't make sense. But most of the time, it should make sense to you. If you go ahead and prepare yourself each week for the material that you covered last week, by the time that is going to come for your lecture exams, you have already reviewed the material once before. All you have to do is that go ahead and look at the material one more time, and you're ready to come and take the exam. It's a smart way of studying. Is a good student. This is, what do you think? You think that good students come with, I had a student come into my office and say, Dr. Nasser, I am not just smart. I said, that's baloney. Don't come on with that kind of thing to my office and talk about you're not smart. That's nonsense. I think that you might be lazy, but you're not, you're as smart as everybody else. Because of the fact that you are all intelligent. You got to believe in yourself that you are intelligent. But you're going to succeed if you want to succeed in this class. If you are just here because your parents forced you to be here, that's not going to be a good start. You're here because you want to succeed. You want to learn. And I will promise you, you will learn. But that commitment going to come from you. So make sure that you prepare yourself for the quizzes. That's going to prepare you for the exam. That's going to go and ensure that you will succeed. One more thing I want to point out. Again, everything that I'm saying, because I have experience in previous semesters, so I have been teaching for this is my 12th year of teaching in university. I'm new here, this is my second year in Dominguez Hills, but I was a professor in Kent State, Ohio for about 10 years, so this is my 12th year of teaching. If these things have happened to me before, I had a student coming to me last semester telling me that they have got 69.8 and they were asking to be able to go ahead and get a C or C minus in their class. I'm not going to give it to you. 69.8 and falling here is going to be D plus, and that's what's going to be. Okay, that's a little bit tough. So how would be able someone try to prevent this painful outcome? Well, if you do what you're supposed to do when you're supposed to do it, you will never be in that position. You might come to my office because you got A minus, you want to get an A, but you're not going to be struggling to be able to survive. So I am going to go ahead and stick to this. I don't like to use the word, but religiously, but I will not change. You are going to earn your grade. I'm not going to give you grade. You earn your own grade. Don't put yourself in a position that at the end of the semester you need a fraction of the point to be able to move on with your life. Make sure you get a head start. That's all going to be it. Get a head start. Okay. Exam format, I will provide you more information, but any kind of questions, mostly multiple choice, some fill in the blank, some true false. I give you a combination of different questions, pathways and things like that. So, but as we get closer to these exams, I will provide you with more information, folks. Reading assignment. I don't know about you, but hey, I remember the time that I was a student very, very well. And what happened was that once I had decided this wild idea of learning how to speak French. So I went and sat and registered in this course, and I went and I sat there in the class, and the teacher, from the moment that she entered the class, she started speaking French, and I don't know a word of French. I felt so bad. It was such a terrible experience. I went and dropped the course because of the fact that I just felt so bad about it. I understand. If you don't understand what is going on in here when we are talking, it's frustrating. But what is the solution? Let's focus on the solution, not the problem. The solution is that prepare yourself. How can you prepare yourself? It's always going to help you if you look at the material that we're going to go ahead and cover. Just look at them. I'm not telling you to sit down and read them in detail. I'm just saying that to go ahead and look at them. So you know we're going to talk about kidney today, we're going to talk about white blood cells today, we're going to talk about red blood cells. Prepare yourself mentally. Now I can give you the explanation behind it because my PhD is in neuroscience. I work on human brain, I work on human diseases, the diseases of the brain, so I know how memory develops. And I'm telling you, if you look at the material before you come to the class, just glance at it, you're going to get a better grasp of the material than if you don't. However, I have also laboratory exercises for you on Blackboard. So each week you come to the laboratory, everything is already set. You got lab 1, lab 2, lab 3. Look at the labs before you come to the class. Don't come to the class when you're completely clueless. It's going to make you confused. By the time that you're going to see what's going on, it's time to go, and you're going to miss the opportunity to learn. So try to just look at these things. Just approach this with an adult mind of trying to go and get the best outcome out of this thing. Okay? If you're going to go on a 1,000-mile trip and you haven't changed the oil in your car for 6 months, how many of you think it's a good idea to go ahead and change the oil before you go on a 1,000-mile trip? It's common sense. It's not something fancy. I'm not just being mean here. Because of the fact that if you see that I'm very passionate about this material that I'm telling you, because nothing is going to bother me than when I see you guys fail. Because I don't see your failure as your failure. I do see your failure as my failure. And I can tell you one thing, I don't like to fail. And I will do anything not to fail. But again, you're going to earn your grade. You're going to earn your way out of here. And it's going to be your responsibility. I will help you. So please make sure that you prepare yourself. You prepare yourself by looking at the material that is available. I have files there, video files. Every week I have divided them. I want you to look at those files before you come to the class. Now some of those files you might want to look at it more than once, that's fine. If you do it, it's going to help you a lot. So if anybody is having a problem, you got to do these steps. I'm sure that the problem is going to subside. It's going to come down. It's not going to be as bad. Okay, I'm just describing how much of your quiz is going to work. By the way, I will give you extra quizzes at the end of, by the time that the semester is going to be, so the total points of the quiz is going to be 100 points. Each quiz is going to be 10 points. So if I give you 10 quizzes, it's going to be 100 points and that's it. I will give you more than 10 quizzes, okay? I usually, last semester I gave 12 quizzes. So if everybody got the quizzes right, they would have 120 points. The question is that what's going to happen to that 20 points? I will add it to your grade, okay? I will add that to your grade. That's going to be a lot of help for those of you who are really striving for a high grade or passing this course, whatever it's going to be. So make sure that you use those opportunities. Now, there is not going to be any makeup for the laboratory exam or any makeup for the quizzes. If you miss the quiz, you miss the quiz. I'm not going to give you makeup. Again, same story. We are having more than 10 quizzes. So if you miss one, you're still going to have opportunity to get your fair share of the other 11 quizzes that I'm going to go ahead and give you. So be aware of that. These points, you might not do well on your first quiz. I understand that because you don't know what kind of question I'm going to go ahead and give you. However, after the first or second quiz, you should be in command of the material. You should be able to go ahead and really nail these quizzes down because they're going to accumulate. They're going to accumulate and they're going to help you at the end of the semester. But yes, I will add that to your final grade. Anything extra. If you are, and I will tell you this again, you're going to have slide boxes and microscopes in the laboratory. Every 2 people in the class are going to be responsible for 1 microscope and 1 slide box. If at the end of the semester, now, accidents happen, things break, I understand that. That's not my problem. You need to let me know. If at the end of the semester, if I go through your box and through your microscope and I see things are not the way that they're supposed to be, I will take off points from you. Okay? I will take off points from you. You're responsible for that box, you're responsible for that microscope, and I will get to the bottom of the problem, what has happened, or one of the 2 people, which one of the 2 people has done this, and I will take off points from you. So make sure that you take care of your belonging. Make sure that you take care of your slides. Make sure that you take care of your microscope. Again, there is not going to be any makeup quiz or laboratory exams. You have to let me know what's going on, then we decide accordingly. That depends on what your excuse is. I might let you go ahead and take another exam. It's not going to be the same exam as other people, but I might not allow you if your reason is not going to be valid. Add and drop, withdraw. The deadlines are there. You would be responsible to keep up with the dates. I had a student last semester again coming to me 3 days after the deadline was passed, and he wanted to go ahead and drop the course. I cannot do anything for you, okay? You got to keep up with these things, okay? Students with disability. People, if you have a disability, we are here to help you. We are here to accommodate you, but there is a procedure, and that procedure is that you got to go ahead and speak to these student disability officers, and they will draft a letter for me, and they will send it to me, and I will accommodate that. And nothing is going to go ahead and count against you. Okay? Don't even think about that. But there is a procedure that needs to be followed in order to get things done right. So please make sure that you go ahead and talk to the student disabilities, and they will send me a letter, and I will accommodate you. Okay? No problem. Pregnancy, if somebody is pregnant, just let me know, because sometimes you're going to go in and use, reagents and things like that. There is nothing dangerous, in that we're going to go, danger is a relative term, Yeah. I was reading the news that in Tennessee, a couple kids drank methanol and they died. And methanol they use in the laboratory. So the thing is that I was thinking about that, methanol is not dangerous. No, it can be very dangerous. But the idea is that if somebody is pregnant, I would like to take extra caution. Okay, I am just that's one of those. If you see me driving down the highway, speed limit is 65, believe me, I'm driving 65. Okay, I'm that type of person. I don't want anybody in danger in my class. I want to protect you. I will protect you with my life, and I want to make sure that, you know, if you're pregnant, let me know so we just take any extra precautions. For example, you're going to go ahead and draw your blood. You're going to bleed today when you come to the laboratory if you have not already bled on Tuesday. If you were pregnant, I wouldn't bleed you. So that's type of things that I want to know so I would prevent from happening. All right, weather condition. If the school is closed, the class is closed. Okay, so don't come. Okay, I am at home, I'm in my bed, and I'm not going to be here. Even we have an exam, we reschedule, so don't worry about it. Student code of conduct. Every student has a responsibility, every faculty has a responsibility. I know my responsibilities, but the question is that do you as a student know your responsibilities? If you want to know it, you can go to that link and you would be able to see what is going to be the student code of conduct. Dress code, I told you about the lab coats. Electronic devices, you can go in, again, I am recording everything, but if you would like to go ahead and record your own lecture, because some people like to do it, they can go ahead and upload it in their car and listen to my beautiful voice when they're driving home. You can bring this. I have no problem with recording anything. Okay, you can go ahead and record the lectures if you want to. You can bring your computer and your iPads, you know, and all this surf and things like that. That's quite all right, but keep it for the class. Keep it for the business of the class. After that, after the class, do whatever you want to do. But when you're in the class, please keep it professional, because there is nothing that I despise more than coming and confronting you and asking you to leave the class because you're watching a movie on, you know. And I have done that. Believe me, I have seen, in my 12 years, I have seen so many things. Let me tell you one that most of you think that is not going to be possible, because it was not here. I don't see it happening in the Dominguez Hills. I was asked to give a guest lecture for a biology course. And it was in a big auditorium. I mean, I'm talking about auditorium of 150 students, you know, sitting in that class. And I'm talking about human brain. And I go ahead and give the lecture, and I turn around in the middle of my lecture, and I see there are two. There's a girl and a boy in the back of the class, and they are at it. They are kissing, and I never seen anything like that in my life. I had to stop the lecture. I said, you two come to me. I said, this is the door. I said, you leave this class and do not come back as long as I'm here. I don't get enjoyment out of that. I am here to tell you about the brain, how the brain works. I'm not here to confront you over stupid things like that. So please, be considerate, be professional. This is a professional program. I want everybody to be professional in this sense, okay? I am not that uptight, and I just want everybody to understand what is going on in this class on the first day. Because things are going to, you know, mellow down as we go through the course. As I be able to attract your attention, things are going to work out much better. I just want everybody to understand what is the situation here. I'm not trying to be mean. I'm not trying to scare anybody. I'm just trying to go in and give you a realistic perspective about what is coming up for you. Okay? So that's my only intention. So please be professional in the course. Yes, you need to have some understanding of the computer science, you know, how to turn on the PowerPoint, how to be able to use PDF, simple stuff. Many of you are not going to have a problem, but every once in a while somebody's going to come who has not had any experience. It's okay. If you cannot get enough, you know, help, I am always available for you. Come and talk to me and I will help you. Professionalism. Again, I understand life is hard. I do understand. Nobody understands this as much as I do. But the bottom line is that if you have a child who is sick at home, okay, and you are waiting for a babysitter to text you if the fever goes 105, 104, and you have to go home as soon as possible, put your cell phone on a vibrate. If you get the text, get up, leave the class. I'm not going to have a problem with that. Take care of the business. If you have to leave, leave. But don't answer the phone in the class. I have seen it being done. Okay? Do not do that, because I'm not going to respond well to it. So the thing is that common sense is really a good thing for us. If you be able to follow common sense, go ahead and take care of your business, come back, and everything's going to be fine. I got no problem with that. All right, there are some important dates here. Final exam is up there. You know, any one of these dates might be changed, so it's all going to be depending on how we progress in the course. This is the tentative schedule, which means that we are not going to be able to keep up with this schedule, but we try. That's all matters. We're going to try to keep up with this schedule, okay? Why are you not going to be able to keep up with the schedule? Because it's you guys that are going to go in and set the pace of the course. I will not leave, or I am not one of those people who try to cover certain material regardless whether or not people understand it or not. Okay? I can look at your eyes and I can understand whether or not you understand what I'm talking about. Okay? I want everybody to understand that this is a basic concept, to answer questions. So if you guys ask a lot of questions, I am going to go slower. If you don't ask questions, to me that means that you understand everything. I'm going to go much faster. Okay? You are going to set the pace of this course. So ask questions if you want me to go slower. Don't ask questions if you want me to go faster. There are dates of the exams that I have put up here. But I want you to know that the way that I give the exams, I have to cover certain topics. So if by that date I am unable to go ahead and cover, for instance, RBC morphology, I might have postponed the exam 1 week or 1 day or something like that. I might change the date. That's quite all right. That's what we call this a tentative schedule. Okay? But I will always give you head notice so you won't be surprised. And this is going to be the tentative schedule for the laboratory. This one we're going to stick to it because each lab is divided to one exercise, and I designed the exercises myself, so we would be able to go in and complete all the exercises during one lab. So we very likely stick to this schedule, and we would be able to complete it. All right. Now, what I am going to go ahead and do is that I am going to go ahead and give you a sheet of paper. I'm going to pass it down into bundles. What does this sheet of paper is going to tell you? This is just mutual agreement, I call it. Take one and pass it down, please. After you read the syllabus and you have understood the syllabus, what I want you to do is that next week when you come to your laboratory, corresponding laboratory, Tuesday or Thursday, either one, you need to write your name, you need to sign your name, and you need to go in and give that sheet to me. I will put in your folder. Okay? All it says that you read the syllabus and you understood it. Okay? Don't be surprised when you come next week, I give you a question from your syllabus. Don't be surprised. Remember, everything that we covered the week before, we covered the syllabus here, you will get questions from the syllabus. I just want to make sure that everybody understands if at the end of the semester you come and say, I'm sorry, Dr. Nasser, I didn't know that. I am going to take out this pen and paper and I say, this is your name, this is your signature, you should have known that. Okay? That's the purpose of it. Bring it to me when? When are you going to bring this to me? In the laboratory. In the laboratory next week. All right. Now, without taking any more time off the class, is there anybody who have any questions right now that I would be able to answer? I don't know whether or not I would be able to answer, but do you have any questions? My question is the lab start today? Today? What time is the lab is going to start today? 3:30. Check the syllabus. For quiz, I do different things. Sometimes it's going to be short answer, sometimes it's going to be fill in the blank, sometimes it's going to be diagram, sometimes it's going to be multiple choice. As long as you're going to know the material, you're not going to have a problem answering. If you're trying to memorize in this class, I got bad news for you. Memorization a little bit is good, but if you are placing all your eggs in one basket called memorization, you're going to have a hard time. You've got to comprehend the material in this class. I will give you a lot of case studies. I'll give you patient cases. I'll give you patient cases that are real. These are people who were sick in the hospitals. And you have to be able to solve them in order to be able to answer. So it's not going to be A, B, C, D, this, that, just memorization. I'm not going to give you questions like that. You can talk to the students that I have had previous semesters, and they will go and testify to that. You got to comprehend the material, and I'll emphasize what that means as I go through the lecture. Okay? I emphasize it. You comprehend it, or you're trying to memorize it. I will tell you these things. So I will help you out to understand what kind of question is going to be coming to you next week. And next week, the first one always going to be fun, because you have no idea what's going to be there. So you're going to get it. But again, you know the material, you're not going to have a problem any type of question I give you. Other questions?
Do we need Scantrons?
Do you need what?
Scantrons?
Scantron. Thank you very much. That's a good one. Yes, not for quizzes. For your exams, for your lecture exams. So you need 4 Scantron sheets throughout this semester. For exam 1, 2, 3, and the final exam. Yes, you need Scantrons, yes. Not for the quizzes. I will give you the sheet just like this for the quizzes. Good question. Thank you very much. See, things like this come, I didn't think about that, but yes, you needed Scantron for your exam, but not for the quizzes. Other questions? Other good questions? Please.
You said that we could find our hematology book online. Can we also find the lab?
Yes, Atlas is there. So look on the blackboard, on the left panel, on top, I have the links. You can go there. Yeah, sure. Other questions? Are we ready to get started on this journey? I want to hear some emotion in here. Yeah. You think it's easy for me to come and stand here and try to go ahead and entertain you? Because I will try to entertain you. Because science learning is supposed to be fun. If somebody tells you, it's dry cut, I am not that type of person. I will make it fun because these are going to be real stuff that you're going to be applying it in the patient cases, patient situations. You're going to learn. Everybody in here might have a family member or know somebody who has leukemia. Don't you want to be able to help these people? Haven't you seen when somebody goes to the doctor and they have no idea about medicine or cell or biology? They're completely lost. They are at the mercy of the doctor. Now the doctor is talking to you, but he's actually thinking about his yacht that he's going to go in and purchase. Now what? Don't you want to be a person that is being able to advocate for your family member, for your friends, for the patient? Well, you need knowledge. Okay? And this is the step that you would be able to acquire that knowledge about hematology. I will help you to get that. Other questions? All right, guys. I am really, and I use this, and somebody told me that I should say that too. And I say guys, I mean guys and gals. Just something that comes out of my mouth. I'm not just talking to the guys, I'm just talking to everybody, okay? So don't get, a lady came to my, after I was done with the lecture, said, Dr. Nasser, why you just called guys? You're gals. I said, oh, come on. I just said speech, I don't mean anything by that, okay? We are ready then. Okay, let's get started. We're going to talk about hematology. The word heme means blood. Ology means the study of blood. Hematology, study of blood. Ology means the study of blood. So, what is blood? Can anybody tell me what kind of tissue is blood? Connective tissue. How many types of tissue do we have in our body? How many? 4. So we just talked about connective. What are other type of tissues we have? Epithelium? What's that? Skeleton is part of the connective as well.
Ligament?
Cardiac.
That's connective too. Muscular. Is that what you're saying? The cardiac is part of the muscular. So you got muscular tissue, you got connective tissue, there are a couple more. Nervous tissue, exactly, nervous tissue. You got nervous tissue, so we got cardiac, we got epithelial, we got nervous, and we got connective. Connective tissue is the weirdest one of all. Because can anybody in a simple word tell me what was the general characteristic of a connective tissue? If I go ahead and put 4 pictures in front of you, and I tell you pick the connective tissue out of these, what would you look for? What you're going to look for is this. Remember, in the case of connective tissues, cells are separate from each other. For example, think about epithelial tissue. One cell is attached to another cell. This is not the way that the connective tissue is going to be. Connective tissue is going to consist of these cells, and these cells are going to differ depending on what type of connective tissue we're talking about. And in between we have something. What is that? You got to speak up. This is called what we used to call as matrix. Now, if your matrix is going to contain calcium phosphate, you're going to call that thing bone. Okay? If your matrix is going to consist of glucosamine, chondroitin, you're going to call it cartilage. If that matrix is going to consist of plasma, we are going to call it blood. Okay? And that's very interesting about the connective tissue because it's going to have different consistencies. Bone is hard. Cartilage is malleable. Blood is fluid. Okay? And that's wonderful, but it's going to connect different parts of the body to one another. So we're going to talk about a little bit about the history, and we're going to go in and get an introduction. This is going to be an introduction, and just get you ready to get busy with this course. I already told you, how much blood do we have? About 5 liters. Is this true that males have more blood than females? Yes or no? Yes. Who said yes? Are you a chauvinist? No, you are not. Then why is that males have more blood than females? What is the reason behind it? Everything in science has a reason. What is the reason?
Is it because they have more muscle mass?
More muscle mass. Males on average are going to have more muscle. If you have more muscle, you are going to have more blood supply. If you have more blood supply, you are going to have a larger volume of blood. Otherwise, don't miss. If you are having a small guy who is petite and a larger female who is larger, that doesn't mean that this petite guy is going to have more blood than the female. It's a matter of muscle mass. In general, males have more muscle mass. Therefore, that's why they have higher volume of the blood. Otherwise, it's all going to come down to this muscle mass. Blood does a lot of things. A lot of things. But the question is that if you are trying to prioritize the function of the blood, how would you prioritize? What is the primary function of blood? Transport oxygen. The reason we are prioritizing because if that oxygen, that flow stops, you're gone. You're going to go ahead and die. So yes, it's going to be able to carry oxygen from the lungs to the tissue. I want somebody to tell me, why do we need oxygen? What's that? Exactly. You need oxygen to go in and make ATP. You folks remember mitochondria? Mitochondria, aerobic respiration. Aerobic is going to indicate you need oxygen. If you're drowning in the water and you die, it's because of the fact that you couldn't get the oxygen to your cells. Mitochondria, respiratory system stops working. You stop making ATP, and ATP is gone, you will die. Okay? That's what death is, when you're not producing any more ATP. And that's what's going to happen in this case. So what's going to happen here is that oxygen is going to be transported from lungs, it's going to go to the tissue, and at the level of the tissue, what's going to happen after you produce the ATP? Is there going to be any waste products? What is that? CO2, carbon dioxide. You got to get rid of. What is going to happen to you if one day all the people go on strike? People who are sanitation, people who come and take your garbage out of the garbage bin. If they all go on a strike for a month, what's going to happen to you? Garbage is going to build up. With garbage is going to be disease. That's what happens with people who are going to have kidney problems. They are unable to get rid of the waste products, so the waste products are going to accumulate and they're going to cause disease. Can anybody tell me what a disease is? What's the definition of the disease?
Disruption of homeostasis.
Bravo. I couldn't put it any better. That's perfect explanation. Any disruption in homeostasis. So people, body function is not engraved into cement. You have ranges. Whenever you guys get into the real world and you are doing these experiments for patients and things like that, you will see, for example, if you're doing patient sodium, there is not one number, there's a range of number. If I go ahead and look at your hemoglobin, and your hemoglobin is reported as, for example, 14 grams per liter, what's going to happen here is that in front of this number, you will have a range, 12 to 16. What that is referring to is that, first of all, not everybody is going to be specifically the same number. There is this range. This is homeostasis. You are gradually, for example, females who are having menstrual, they go through the menstruation, their hemoglobin might become 13 at the time of menstruation. But quickly they would be able to recover and go back to 14 or 14 and a half. So this is continuously changing. That's what homeostasis is, to maintaining a stable environment. Okay? Stable environment. All right. We talked about the plasma. And we talk about cellular structures. I like to call them elements. I don't want to call them cells, because one of those guys up there is actually not a cell. It's a cell fragment. There are 3 groups of cellular elements that we're going to get to know in this course very well. We're going to start with red blood cells. Then we're going to move into the white blood cells. And if we have time, we're going to complete the journey with platelets. Platelets are not cells. Platelets are cell fragments. And I will tell you more about it in a few minutes. Little bit history. People who don't learn from history, they are doomed to repeat it. And that's something that completely amazes me. Just completely amazes me. It's just something that, I see with my eyes and I cannot believe. I just cannot believe. I cannot believe that, for example, 2008, the economy was collapsing when we got the new president coming in and saving this country from the bottom abyss. And today, when you hear the news, you turn on the news, everybody's talking, oh, how wonderful things were in 2008. Okay? Because they don't learn from history. We need to learn from history. Okay? You think that it's difficult for you? You should have been, instead of the Kircher. Kircher was in 17th century. He was a priest. At that time, science was not expanded the way it is now. What it was that Kircher was a geologist, Kircher was a doctor, Kircher was a biologist, Kircher was, working on plants. He would write about religion, he would write about philosophy, he would do a lot of things. But one of the things that he did was that, I think that he was one of the first people that was able to correlate. You know what bubonic plague is, right? Bubonic plague, the plague, when they are talking about bubonic plague. Yes. It is devastating, it's going to kill people, it's going to kill 3 out of every 4 people who are contracting disease. Bubonic plague is due to what bacteria? Yersinia pestis. That's good. That's good. He was the one. First, they were saying that, at that time, they were saying that, these people have done, they did not, go to church, or they didn't do this, they didn't do that, they're all being, punished by God. Kircher, he was a priest himself. He came and said these little worms inside of the blood, because he used those rudimentary microscopes and Many people say that he saw the white blood cells and he thought it's the Yersinia pestis. But whatever it was, he correlated this element inside of the blood to the disease. That's 17th century. Okay? When you are looking at the Leeuwenhoek is going to be the person that many people contribute the discovery or creating the microscope, but it's incorrect. There are other people who discovered microscope first before him. And he was the one who was a glass grinder. He was lens grinder. If you wanted to get glasses, you would go in and meet Leeuwenhoek. And he was able to improve on the microscopes. And believe it or not, he is the microscopes that you guys are going to be using in the laboratory, basically the same model that he developed. 17th century. Okay? Not a big deal, but he was able to go and look at the red blood cells. You in the laboratory are going to be able to go up to 1,000 magnification. So you're going to have an image, you're going to be able to expand it to 1,000 times. He was able to do it with those rudimentary instruments up to 200 times. But he was able to describe the red blood cells. He was able to go and describe the white blood cells. Bizzozero was an Italian physician. Italian physician who was using the fruit of these people's labor, and he was able to go ahead and look at the blood smears, and he was able to identify platelets. And he correctly identified the function of the platelets in blood clotting. That was this gentleman in the 19th century. And James Wright, beginning of the 20th century, when you guys go to the laboratory, you're going to make a slide of blood today, smear of blood today. Next time you come, you're going to stain that. And you're going to stain it with the same technique that this guy discovered more than 115 years ago, 110 years ago. That's what he did. Because in order to be able to visualize these small images on the microscope, you have to color them differently. For example, if you are going to have DNA, do you think DNA is acidic or basic? Acidic. Is there anybody have a question about that? I want you to think about what is the DNA stand for. Deoxyribonucleic acid. So if you use a basic dye, it's going to stick to this DNA and it's going to make your nucleus look what we call basophilic or deep blue. And if you have something such as lysosomes that are going to be very acidic, you're going to be able to stain it with the same thing. If you have something that's going to be very basic, you're going to be able to use acidic stain and make that look different. Different structures in the cells is going to look differently. You will see that in the laboratory. This is the guy. 1900, okay? So that is the history where we started moving forward into, and there are different aspects of the history. For example, when you're talking about blood transfusion, ADO blood typing, that has its own history. I don't want to get into it. I hope that your blood bank, teacher be able to go and tell you about it. But I'm just telling you there have been a lot of trial and error. A lot of people died in order to understand and learn what we know today. Don't ever forget that. You got to learn from the mistakes of others. Okay, I got a question. You folks know that every cell, well, let me ask you this. How does a cell decide to do what? How does a cell that makes my hair is going to know that it's supposed to become a hair? How does a skin cell know that it's going to become skin? How does a cell in my brain is going to know it's going to become a neuron? What is the key? Gene expression. Okay? We all came from sperm and an egg. They fused together, they fertilized each other, they became what we call a zygote. Then the zygote is going to start dividing until you have a lot of smaller cells, and then you are going to have a different genetic profile, gene expression profile for different cells. One of them is going to become hair, the other one is going to become skin, the other one is going to become a neuron. Okay? So it's going to be important to understand the concept of gene expression. How many genes do we have on average, human beings? Something about 24 to 25,000. That's right. So what's going to happen here is each one of the cells in our body, on average, about 300 of them are going to be active. Okay? On average, 300 genes are going to be active. So when we are talking about the hair cell versus a neuron, the only thing that's going to be different, the DNA sequence is going to be the same, but it is the expression of the genes that's going to differ. We make one neuron and the other one is going to be hair cell. When you hear in the news that they are using stem cells, this is what they are trying to accomplish. The idea is that to be able to get that cell, be able to tell it, you become a neuron, because this person has Alzheimer's disease or Parkinson's disease, we want to use it to transplant in the brain. You become a spinal cord motor neuron, because this person is paralyzed, we want a motor neuron allowing this person to be able to walk again. They are trying to control gene expression. And I promise you, we will get there. I can assure you within the next 5 years, type 1 diabetes, we're going to start transplanting stem cells in people who have type 1 diabetes, and we're going to cure that disease. We're not going to treat the symptoms, we're going to cure it. You are at the junction that things are happening so fast. You guys are going to have this vista of creativity coming out to you. I wish I could be your age, okay? Because I really want to see 20 years down the road how this science is going to go change the world. Yeah, of course, if we don't destroy ourselves, because sometimes I think that we might do that. But if we be able to keep ourselves, in the game, we are going places, people. You are at the junction at a very good time. Very good time. Okay, saying all that, what was the purpose for me to say that? Everybody understand every cell needs DNA? Yes? This is a red blood cell. Where is the nucleus? Isn't that where the DNA is? Where is the DNA? In the red blood cell. How does the red blood cell know what it's supposed to do? What's that? No. Hemoglobin is a mailbox. It's a mailbox. You put oxygen in it, delivers it, and comes back and picks up another one. But doesn't know what to do. A lot of things happening inside the red blood cell. How? Where is the information coming? They do have nucleus. But you got to know that red blood cells are produced all the blood cells are going to be produced in the bone marrow. They do have a nucleus when they're in the bone marrow. But when they mature and they're going to come out of the bone marrow into the blood circulation, they will lose their nucleus. And they're going to come in. How is that possible? They are losing nucleus and they're coming to the circulation. I am confused. What's going on in here? Well, I'll tell you what's going on in here. Everything that needs to be inside of these red blood cells, by the time they mature, is produced. It's there. Hemoglobin, each red blood cell has a lot of things, but one of the major things in the hemoglobin is going to, inside of red blood cells is going to be hemoglobin. Each red blood cell has on average about 200 to 300 million hemoglobin molecule in it. All that was produced when it was in the bone marrow, when it was mature. Okay? When they lose their nucleus, then they are matured, they can come out of the bone marrow, they're going to be able to do their jobs perfectly. But when you go to the laboratory, you do see, I have slides on in your boxes that you will see nucleated, think about it, nucleated red blood cells. You see it in the circulation. What does that tell you? If you see your child has nucleated red blood cell in their blood, and we are talking about 10-year-old kid, 10-year-old boy has nucleated red blood cell. Don't get emotional. Look at it logically. What does it mean? Bone marrow problem? Not necessarily. No. Yes, yes. To the extent, yes. What it's going to be the problem is going to be this. Common sense. There is a need for red blood cells. There is a shortage of red blood cell. Your bone marrow has to kick in immaturely into the circulation because even though they're going to be nucleated, they're still going to have hemoglobin in order to be able to carry a little bit of oxygen. They're not going to be able to be 100% efficient, but they're going to be 20% efficient. It's better than 0. So the bone marrow is going to kick him out prematurely. Okay? Now, you can have bone marrow problems for that, but many times, what if you're anemic? What if you are going to be a person who is 50 years old, you just got your heart valves replaced. You got a mechanical heart valve because your other original valves got calcified, they don't close and open, you have to replace them. Now, one of the things that's going to come with the new heart valves is that as the red blood cells are going to hit the heart valves, they're going to rupture. You're going to have hemolysis. Hemolysis when the red blood cells break down. Now, if this person's red blood cells is breaking down at a very high rate, he's going to become anemic. What's going to happen here is that when he becomes anemic, in his kidney, he has this mechanism that's going to be able to detect the amount of oxygen in the blood. You following me? Is everybody following me what the situation is? This is comprehension. Okay? Don't be surprised if I give you a similar case study on your quiz. These are the type of questions that I want you to understand. You got to be able to find your way out. Okay? Person got their heart valve, causes hemolysis. They lose blood because red blood cells are rupturing. Hemoglobin is soluble. If you have too much of it, it's going to go through the kidney and you're going to urinate it out. You're going to lose that hemoglobin. Your kidneys have the machinery in order to detect low oxygen in the blood. Low red blood cells means low oxygen. And they in turn are going to produce a hormone. Tell me. What's that hormone? Erythropoietin, or some people call it EPO, whatever. Erythropoietin. You know what erythropoietin is going to go in and do? It's a hormone. Hormones travel through blood. It's going to go to the bone marrow. You have a stem cell in the bone marrow. That stem cell can become a white blood cell. That stem cell can have the capacity of becoming a red blood cell, and that stem cell has the capacity of becoming a platelet. Erythropoietin is going to ascertain that this red blood cell goes this direction, becomes a red blood cell. Not white blood cells, not platelets. Beautiful. Your body is able to take care of itself. Okay? Now, if you are going to be in a high demand, your bone marrow is going to kick out immature cells. You're going to see nucleated red blood cells. Now, sometimes it's not concerning. If I see a nucleated red blood cell in somebody's blood smear who is 50 years old, I will be very concerned. I will think about bone marrow disease. I will think about the worst case. But if I have a newborn who was just born 2 weeks ago, and I see nucleated red blood cells, I wouldn't be that much concerned. You know why? Because the liver function, spleen function is not going to kick in its full capacity in newborns, so things are not going to look very normal in those individuals. So you got to know the patient who you are looking at. You got to look at what that slide belongs to. Is it a male? Is it a female? Is it a young person? Is it an old person? It all matters who is on the other side of this slide. You got to always take those things into consideration. Hemoglobin, the size of the red blood cells is about 6 to 8 micron. I want you to remember this, I'm telling you. When the red blood cells are anybody knows what is the lifespan of a red blood cell? 100, on average, is 120 days. So from the time that they are released from the bone marrow into the circulation, they're going to be around about 120 days. And then they are going to be taken out by these cells we call macrophages in the spleen or in the liver, and they're going to be recycled. We will talk about that. We will cover that mechanism completely later on. But the bottom line is going to be that after 120 days, they're going to be recycled. Okay? when the red, this is what I want you to remember. When the red blood cells are going to be younger, it's different than human beings. This is the key that you will always remember this. You don't forget it. When we are humans, we are small. As we get older, we get bigger. Red blood cell is the other way. It's bigger first. As it gets older, it gets smaller. Remember that. That's going to be a big, big hint for you later on. That the younger red blood cells are larger in size. Okay? All right. The size of the red blood cells is 6 to 8 microns. Again, we are giving a range in here. We don't say only 6, only 7. There's a range, okay? You have blood vessels, we call them capillaries in your body, that the diameter is about 4 microns. 4 microns, okay? Think about this. You got a hole about 4 microns, you got a ball about 6 microns, and it's not going to go in if it was not because of this shape of the red blood cells. You can see that the center of the red blood cell is going to be concave. I will tell you more about the beauty, indeed beauty of these red blood simple cells we call red blood cells, for you to understand that without this shape, things go wrong. That's what happens with sickle cell anemia. That's what happens with hemoglobin C. That's what happens with hemoglobin E, because it affects the shape, and if the shape changes, what do you think is going to happen? If something happens for this red blood cell up here to change its shape to something like this, what's going to happen? It's going to block. It's going to get cut in those small blood vessels. And the blood is not going to be able to go distal to that point. Now you understand why people with sickle cell anemia are going to start developing tingling and neuropathies at the end of their fingers and toes. Now you understand why these people lose their fingers. Because of the fact that whenever they get sickle crisis, these are going to go and block the pathway. And if you don't get the blood to those cells, they're going to die. There is no miracle about it. There is no fancy or magic about it. If you don't get the oxygen to those cells, they have a certain short period of life to survive. If you don't get it there, they will die. Okay? And then they die, they have to be removed. So this shape is very important. Remember that. Because you're going to look for this shape when you go to the laboratory. Okay? This is a very important thing. See how beautiful those are. That central pallor on both sides is concave. And I will tell you more about it. Don't start me because I want to finish this lecture. If I get started on that one, we're going to go. We're not going to be able to finish the lecture. So we will talk about this again when we talk about red blood cells. I'm just introducing to you what we're going to do in this semester. Okay, we all have given blood, or we have seen people give blood. Is there anybody who doesn't know what the blood looked like? All right, good. It's red, okay? Maroon red. Depends on if you're looking at arterial blood, is going to be much brighter. The venous blood is going to be darker. And that's going to be all depending on the oxidation stage of iron inside of the hemoglobin. If the oxygen is going to be bind to the hemoglobin, to the iron in the hemoglobin, that hemoglobin is going to be brighter red. If there is no oxygen bound to the hemoglobin, that's going to be darker maroon red. In either case, when you are getting your blood, it's going to be completely red. But you can see in here that the blood is going to be looking a little bit different. Because this blood spun down. It allowed to clot, and then they spun it down. So you're going to have 3 layers. You're going to have the red blood cells. They are the heaviest one in the bottom. Okay? You're going to have the majority part usually is going to be the plasma, the matrix of blood as a connective tissue. And there is another layer here. There are different people call it different things. I learned to call it buffy coat. And that is going to contain white blood cells and platelets in the buffy coat. And those are going to be in here. Sometimes you see it very clearly. Sometimes you might not even notice it. It's so thick. Okay? On the other hand, what you are seeing in the image that is below, down there, that is a special type of stain. Okay? We call it new methylene blue. That's why they look kind of bluish. You do see this cell and this cell in here, they got this speckles, this bluish, dark bluish color inside of the cytoplasm when they are stained with new methylene blue. What new methylene blue is going to be able to do is that it's going to be able to bound to ribosomal RNA. Ribosomal RNA. Can anybody tell me what is the function of ribosomal RNA? Not the detail, just the basic function of ribosomal RNA. What do they do inside of a cell? What's that? Ribosomal RNA. Where do we find ribosomal RNA? What is the function of ribosomal? What is the function of ribosomes? Let me ask you this. I heard that. Protein synthesis. Protein synthesis, that's right. So ribosomes are hybrid molecules. They got a protein component and they got an RNA component. When I tell you ribosomal RNA, I'm talking about the RNA component. So when you treat them with new methylene blue, what's going to happen here is that the stain is going to be bound to the ribosomes, the ribosomal RNA, and it's going to make these distinct speckles inside the red blood cell. What is that all about? Very simple story. See, people, in order, hemoglobin, you all know, is a protein, right? It's a protein. Proteins need to be synthesized from the DNA transcription into the translation into the final product, which is going to be hemoglobin. Everybody understand what I just said? Okay? So the bottom line is that this is going to indicate this cell is a very new, young cell. Because the RNA that was the ribosomes that were produced when it was in the bone marrow, they have not been broken down yet. I can assure you this cell is about 48 hours in its age. 48 hours ago, it was released from the bone marrow. Within that time, maybe 24 to 48 hours. About 1 to 2%, you can go, again, there's a range. The range in my book goes about 2 to 3%, if you want to be very technical. Of the blood cells, red blood cells in your body are going to be these cells. We call them reticulocytes. Reticulocytes. Can anybody tell me what reticulocytes are now? What are the reticulocytes? Young red blood cells. Young red blood cells on average with about 48 hours maximum lifespan. Because as time goes by, this is going to break down. These ribosomes are going to be broken down and they're going to look more like this one. There is no ribosome left. Okay? How can I use this simple information and use it in order to diagnose patients? How can I be able to go ahead and use this simple concept? If I go ahead and give you a reticulocyte of a patient, what do you think that's going to mean? If I tell you Mr. Jones is going to have a reticulocyte that is going to be 6%, how would you be able to interpret that? Think about it before you say anything. Think about what that means. On average, you have about 1 to 2% reticulocytes. I am coming and telling you Mr. Jones has 6% reticulocytes. What's going to be the answer?
Probably he has bleeding, he lost blood, so bone marrow is compensating for
Bone marrow is compensating, kicking out new cells, kicking out your body is trying to protect itself. That's exactly what it is. The bone marrow tries to compensate, kicks out young cells in order to replace what has been lost. That's exactly what it is. So see, some information like that is going to give you tremendous amount of knowledge about what is the matter with the patient. Okay? We talked briefly here about bone marrow disease. If you go ahead and look at the hemoglobin and hematocrit of a patient, which is we're going to talk about in detail, so if you don't know what they are, that's fine. But the amount of the hemoglobin and hematocrit, usually we're going to, for instance, different places, different numbers, but if you are having a hemoglobin normal range from 12 to 14, and you are having a patient, hemoglobin is going to be 8.0, this person is going to be anemic. You're going to go ahead and do reticulocyte count on it, and you find that there is 1% reticulocyte. How would you interpret that? Your bone marrow cannot compensate. Now you got to find out what's the matter with bone marrow. You got pernicious anemia, you got some kind of disease going on that the red blood cells are not being able to survive. That's what you're going to get out of it. And these are the type of the questions you would expect on your exam. I'm not going to go and ask you, what is anemia? I'm not going to ask you, you're not kids, this is not high school class. I am going to ask you things that you got to think. You got to use your head in order to get the answer. It's not fancy. It's not strange. It's not rocket science. It's critical thinking. Okay? It's being able to get the evidence and interpret the evidence. I don't want you guys just learn things. I want you to learn to apply things that you are learning into the real situations. This is the purpose of this class if you don't know it already. All right. I will tell you about indices in a second. We will do these assays. You will check your hemoglobin in the class and things like that later on, so I'm not worried about that one. Now, you remember I told you about James Wright, who came up with the Wright stain? This is what this stain is going to look like, okay? There are two things you're going to be able to get out of this image up here. One, is this cell bigger than this cell? Yeah. Why do you think that's the case? Perhaps this is the younger one. I don't know how well you can see the what we call polychromasia. There is a reddish in here, there is a bluish tinge in here. That's called, in hematology we call that polychromasia. You're going to get to know it, don't worry about it. Two things, the cell is bigger. I told you why that is. The younger cells are going to be bigger in size than the older cells. Polychromasia in the right stain, when you are going to go in and have reticulocytes, they're going to stain a little bit bluish. We call that polychromasia. Do not call that reticulocyte. I know they might be reticulocytes, but you're going to call this polychromasia. But when you are doing this stain for the reticulocytes now, you can call these reticulocytes. Let me tell you why, because I always want to go in and share with you the reason behind things. I don't want you to just follow me because I say so. The reason is that this guy down here might be a reticulocyte. But this reticulocyte might be in the second part of its 48 hours, and majority of the things that are broken down, so you're not going to be able to see the bluish polychromasia that is associated with much younger cells. So you're not going to count this one in, and that would be wrong. Okay? You only consider these reticulocytes when you use the reticulocyte stain. There are different stains. The one that's commonly used is called new methylene blue. Right stain, reticulocytes are going to appear a little bit polychromatic, polychromasia. CBC, complete blood count. Many of you have done this. You will do it if you have not done. You basically draw blood in purple top tube. A purple top tube has EDTA in it. EDTA is going to go and bind to calcium. When it binds to the calcium, calcium is a very important element for blood clotting. If you be able to remove all the calcium from the blood, it doesn't clot. You don't want the blood to clot for this purpose of the CBC. Okay? So what's going to happen here is that you're going to count cells. You're going to give the numbers for the platelets. You want to actually going to count the cells for the platelets. You're going to count the red blood cells. You're going to count the white blood cells. And you're going to have a number with a unit, specific unit. You're going to learn all those things. You're going to indices. Indices are not counted literally. They are calculated values. When you calculate indices, you are using the information that you already have. You do a simple calculation and you get numbers and values that you can use to interpret what's going on with the patient. Let me give you an important one in here, because this is going to be something that's going to be your bread and butter throughout this semester. Let me explain this, what I mean. One of these indices is going to be called MCV. What does it stand for? It's going to stand for mean corpuscular volume, aka It tells you about the size of the red blood cell. It tells you about the size of the red blood cell. The normal range for the, so it's going to tell you about the volume of the red blood cell, how big it is. The normal range is going to be 80 to 100 femtoliter. Femtoliter is a very, very small volume. If you really need to know it, it's going to be 10 to the power of minus 15 liter. So it's a very small volume. It's going to be, that's the normal range. So a normal red blood cell on average is going to be 90 femtoliter. Okay? This is an indice. It is not measured by itself. It's calculated. How do we calculate that? We actually measure the hemoglobin of the patient. We actually measure the hematocrit of the patient. We actually measure the red blood cell of the patient. We actually measure these. Cell number 1, number 2, number 3. But we don't measure MCV. We calculate MCV. How do we, for example, calculate the MCV? And you will have to do all of these things in the laboratory, but let's go ahead and get a head start. You get the hematocrit. You divide it by the red blood cell value. Of course, these are all going to have their own units. This is going to be, per liter. This is going to be percent. You multiply it by 10. The number is going to be MCV. You just did that. You get the MCV of 110. What does that tell you? You got the large red blood cell. Now, what is causing that? We're going to talk all about that one. But right now, you got this very significant information by doing a calculation of the values that you knew, and you got more information. Okay? Science is evidence-based. Science is not wishy-washy because I say so, that is the case. Science is going to do it based on evidence. Now, this person has large red blood cells. Let's find out why that person has large blood cells. A, B, C, D. And this is the way that you're going to work that way to a diagnosis in order to find out what's the matter with the patient. Okay? Indices. There are going to be a lot of them. You got MCV, you got MCH, mean corpuscular hemoglobin. That's going to be talking about the weight of hemoglobin per red blood cell. You got another one called MCHC. That's going to be mean corpuscular hemoglobin concentration. This is about the weight. This is about the concentration. Does it make any difference? Well, let me give you an example so you understand what the story is. So we had a cell there with a large MCV. It was a large cell. Let me draw it up here. This is that cell. MCV is, what did I say, 110, 120? Let's say 120. You got a normal cell. MCV 90. Okay, 90. Now, I am just going to go ahead and depict it, but you all understand. I'm just trying to go ahead and make a point in here. Let's say we got 3 hemoglobin in here. Okay? Just 3. Let's go ahead and give the same number to here. Pay attention. Do these 2 cells have the same MCH? The weight of the hemoglobin inside each cell? Yes. Do these 2 cells have the same MCHC? No. So what is going to be the idea of MCH and MCHC? They're very helpful. But I like MCHC more because it gives me more information. But you got to watch out when you're interpreting MCH as well. It's going to give you the same information, but MCHC is much more reliable to me. Different people have different opinion. It's going to tell you how hypochromic or hyperchromic a cell is going to be. It tells you about the color. When you are looking at the slide under the microscope, the red blood cell have to have certain intensity of reddener. If you are not going to have enough hemoglobin, they're going to be much paler in color. Okay? MCHC is going to pick that up. MCHC runs, if I recall correctly, again, different laboratories might have a little bit different, but runs about something about 32 to 36. And the unit is going to be grams per deciliter. Grams per deciliter. If you are going to have an MCHC of 30, that's going to tell you red blood cells are not going to be, they're hypochromic. They're not going to have the same color as the normal red blood cells. What does that tell you? Yeah, the person has some kind of anemia. They don't have enough hemoglobin. This is the person that's going to have fatigue. This is the person that's going to run 100 meters and is going to run out of breath. This is the person that's going to have all the symptoms of anemia because they don't have enough hemoglobin. And hemoglobin is going to be the molecule that carries the oxygen. Okay? MCHC. You have others as well. You got RDW. RDW stands for red blood cell distribution width. The smaller RDW, I don't remember what it runs. I think it runs about 11 to 16. I'm going to double check that. You know, I usually don't memorize these things, but as you do it more and more, it just gets stuck in your head. If it's going to be within that range, red blood cell population, they are all basically the same range. Their sizes are the same, similar. But all of a sudden, you're going to get RDW of 19. That tells you some of the cells are bigger, some of the cells are smaller. You have a new name you're going to learn. We call that anisocytosis. Anisocytosis. Remember that anisocytosis, if it's present, is going to tell you that there are different population of cells. They're size-wise, okay? Red blood cells, some of them are large, some of them are small. There is a great difference among the average cell size in their width. There's another one called MPV, mean platelet volume. Platelets are supposed to be very small, but sometimes you will see what we call giant platelets. You will see them in the laboratory. MPV is going to provide more information about the average size of the platelets. Okay? So these are all calculated. I just want you to know that these are calculated value. They're not actually measured, they're calculated. Hemoglobin and hematocrit. Hemoglobin concentration, that makes sense. If you be able to go ahead and hemolyze all the red blood cells, then you measure the amount of the hemoglobin inside that volume, you're going to get the hemoglobin concentration. Hematocrit is very simple as well. Hematocrit is going to be the volume of packed red blood cells. Volume of packed red blood cells. Divided by the total volume of that sample, blood sample. Times 100 is going to be a percent. It's going to be a percent. On average, males are going to have higher hematocrit. You already know why that case is going to be, because they have more muscle mass. Females' range is going to be lower. So for example, you might be able to see that the female's normal range is going to run about 30, I'm just throwing this 39 to 43 for instance. But when you look at the males, you're going to see that the range was, for example, 42 to 46. The difference is just because of the difference on average muscle mass of the males versus females. Okay? So hematocrit, let me explain to you one more time quickly so everybody make sure that you understand that, because it's a very critical concept. If I have a blood tube, and this blood tube is going to be up to here, I let it clot, and I spin the blood for 10 minutes, and you're going to have all the red blood cells going down here. Let's say that from down here up to here is going to be 100 equal segments. And let's say that the red blood cells that we're packing the bottom of the tube, they're going to have about, let's say, 35. 35%, 35 divided by 100 times 100, is going to give you 35%. This person is going to have a hematocrit of 35. Is this person anemic? If this was a male or a female in this case? Yes, a simple test. This test is going to cost about, I would say about 5 cents. You got to go in and do this in a laboratory, they're going to charge you $25. But it's a simple test and it's going to give you a lot of information. It's going to tell you about anemia, people. It's not going to tell you what is the cause of anemia. It's just going to tell you there is a problem. Then we have to go in and investigate what the problem is. Is there any questions? Okay, I usually will give you some time, but today because we talk about the syllabus, I'm not going to give you break in between, but I usually give you at least 5 minutes, 10 minutes break in between. So from next time, I will give it to you, so don't freak out. Okay, I already told you a lot of things that I'm having up here. I told you about anisocytosis. I told you about RDW. I told you about MCH, MCHC, and MCV. White blood cells. So white blood cells are the cells that maintain, they do a lot of things, but the primary thing that I can put it in a nutshell is that provide immunity. If you don't have white blood cells, you will not be able to make it, because any simple thing that is going to be inside your body or outside your body is going to be able to harm you and it's going to be able to kill you. They give us immunity. We have different types of white blood cells. We don't have one type of white blood cells, and each one of them is going to be able to be responsible for different functions. There are a certain number of each one of them is present, at least a range of each one of them is going to be present in the blood, but I promise you that by the time that you are going to get out of this class, not today, at the end of the semester, you will be dreaming about those white blood cells. You would knowing it better than your family members. You're going to know what a neutrophil is. You're going to know what a monocyte is. So there are different types of them. There are 5 important classes of them. There are neutrophils. They are monocytes. They are eosinophils. They are basophils. And they are going to be lymphocytes. And of course, each one of them is going to have a different class, subclasses, but we will talk about them in great detail. If somebody is going to have a low white blood cell, it happens sometimes. We call that leukopenia. Okay, leukopenia means low white blood cell count. You can have high blood cell count. If you are having somebody who has, for example, acute myelocytic leukemia, AML, don't be surprised. Normal range of white blood cells is usually runs about, I would say, about 4,500. And I'm just not going to give you units right now, but I'm just going to try to go and convey the concept. It's about 4.5 to 10.5 times 10 to the power of 9 per liter. This is the number of white blood cells. Okay? So what's going to happen here is that sometimes you are going to fall on this side, leukopenia. Sometimes in the case of leukemias, for instance, or in the case of polycythemia, you might go into millions. Instead of 10.5, you might have 1 million. Okay? So you can have all those conditions. But this is the homeostatic range. You are all healthy individuals. If I draw your blood, you're all going to fall within this normal range. If you have leukemia, if you have AML, you're going to be all the way on that side. If you have pernicious anemia, you're all going to be on this side. So it's all going to indicate different things. Okay? These are the guys. Get to know these guys, because you will get to know. Don't worry about it. I will make sure that you get to know. You got the neutrophil on that side. Get this idea that all the nucleus of the cell always has to be round. Get that out of your head. This is the nucleus. Okay? You got also granules. So there are different factors that's going to allow you to identify different cells. One of them is going to be the shape of the nucleus. Another one is going to be the size of the cell. Another is going to be the color of the cytoplasm. Another thing, you see these dots that are going to be in here? Those are called granules. Different cells have different granules. That's the neutrophil. Look at the granules in the eosinophil. Look at the nucleus of the eosinophil. Look at the size of the eosinophil. This is the basophil. Look at the granules of the basophil. They are not the same. People look at these cells in the first when they're inexperienced, and they say, "Hi, they all look the same." Well, find out what the nucleus looks like. What's the size of the cell? What are the granules that are present? And you're going to see the difference. This is called a monocyte. This is basophil, and this one has a round nucleus. It's going to be a lymphocyte, resting lymphocyte. Each one of these cells is going to be responsible to do different things. Okay? And we will talk more about the details of it as we come to it. Now, this big cell, you don't see it in the circulation. This is a huge cell. Sometimes they are so big, if you're really young and have good eyesight, you might be able to look at the blood and you would be able to see that without a microscope. They are that big, okay? It's about 30 times of the red blood cells. Sometimes they have about 18 to 20 nuclei that are here, okay? This is called megakaryocyte. Megakaryocyte is the progenitor, aka the father of or the mother of the platelets. These are platelets. So when I tell you that the platelets are not cells, they're cell fragments, that's what I mean. Their father lives in bone marrow. It's not going to be in the circulation. Depending on the cues that are available in the environment, it's going to pinch up its cytoplasm with all the proteins inside of it, and it's going to release it into the circulation. When it comes to the circulation, we're going to call it platelets. But they're all going to come from here. Okay? Platelets, megakaryocytes. Endothelial cells are going to make the lining of the blood vessels. I want you to understand, first of all, endothelial cells are epithelial cells. Okay? They're epithelial cells. They're actually simple squamous epithelium. You can see how thin they are, and they're a single cell squamous epithelium. They make the lining of the blood cell. Okay? And they're going to have a very important function. What kind of function is going to be is that everything that's going to be in the blood has to leave the blood vessels and go to the cells, to the tissue. And everything that needs to leave the cells has to come to the blood. They have to cross this barrier of endothelial cells. So these endothelial cells have a lot of functions. They have a lot of function allowing things to go through. They have a lot of functions in coagulation and bleeding stoppage. They have a lot of function. But I want you to look at this electron micrograph. This is not light microscope. This is electron microscope. Look at this. Next time when you are going to go and have an ingrown nail, or you are having a thorn in your finger, you're going to get inflammation, you're going to get swelling, you're going to get pain at the tip of your finger. This is what's happening. You're having a bacteria coming in here. And when the bacteria comes in, all the cells in here, including endothelial cells, they're going to release these chemicals. These chemicals that is asking for white blood cell help. Hey, there is somebody here, you guys got to come and take care of it. If somebody breaks in your house, what do you do? Shoot them down. If somebody's trying to break in your house, not in your house, it's a good idea to pick up the phone and call 911 to get help, okay? Those help are going to arrive, but how are they going to get from here to here? Look at this. The door is in here. Let me tell you something. I usually don't say this in the beginning of the class, but when you're having allergies, some people have allergies. One of the symptoms of allergy, what is it? Your eyes, the tears coming out of your eyes, you're sniffling, your nose is running, right? Itching, and you know why? Because something you were exposed. Now it can be something unimportant, such as the hay fever, the pollen, or something like that. But it triggers the same pathway. When that pathway is going to be triggered, what's going to happen here is that these cells are going to come. These are the guys. Basophils. You see these black granules inside of the cytoplasm. Those are filled with histamine and heparin. Does that make sense now? When you are having allergy, you go to the CVS or you go to the pharmacy. What do you get? Antihistamine, in order to neutralize that. You know why? Because of the fact that when you release the histamine, you see this tiny gap between these two? It's going to open up. It's going to open up so wide that the white blood cell easily can go from in and out of the tissue. With that is going to come what? Plasma. Right? Plasma is going to come out. That's where the swelling is going to come from. Okay? That's where the swelling, because the plasma is going to come in, all these white blood cells are going to come and trigger inflammatory response. So when you go in and take the anti-inflammatories, the antihistamines, what that idea is going to do is going to neutralize the histamine in the basophils. It's going to close the gap, so the fluid is not going to come in, and that's going to relieve your runny nose or itchy eyes. Everything makes sense in science. Hemostasis. This is not homeostasis. Don't make any sense. This is hemostasis. It means blood stoppage. When you're bleeding, you stop bleeding. There are cases such as hemophilia in which there is a problem with the system. You bleed until you die. If you are not intervened, you're going to die. But hemostasis is a fascinating, beautiful mechanism. But platelets are the main player. Platelets are going to be very important in hemostasis. You see these things in here? This is a platelet. This is a platelet. This is a platelet. These are red blood cells that are captured in the network of these platelets and these strands. These strands are called fibrin. It's a protein. Your liver produces a protein that is called fibrinogen. Fibrinogen is soluble in the plasma. So if I get your blood and I spin your blood and I'm looking at your plasma, I'm not going to go in and see fibrinogen. But whenever you injure your blood vessels, you're going to have a bleeding, there is going to be something happening, especially those endothelial cells that I showed you. Those are going to trigger that the fibrinogen, which is a protein, is going to get chopped off, and what's going to be the product of it is going to be what we call fibrin. It's not going to be soluble, it's going to come out of the solution, and it's going to be bound to the platelets. Okay? It's going to create this network that's going to be able to capture the red blood cells and everything else and start the process of healing. Now, if you have a really nasty scab, you can see that sometimes from that scab you're going to start oozing a little bit clear fluid. You know what I'm talking about? You understand now why that clear fluid is not red. Because the red blood cells cannot pass this network. But the water is going to be able to come out. And that's where the clear oozing, that's clear oozing is going to be. It's just the process of healing has started. It has not been completed. Okay? What's going to happen is that the platelets have things in them, have proteins in them. Proteins such as actin and myosin. Have you heard of actin and myosin? Where did you hear about it? Muscle. That's exactly what's going to happen. If you're having that wound in here, if this is going to be your wound, now you are going to have this platelets bound to the wound everywhere, and the fibrin is going to be creating this network. Then the actin and myosin kick in. They do what they do. They contract. As they're going to contract, they're going to start gradually bringing the edges of the wound together, and they start the process of healing. Not only stop it, but they're going to start healing, because you get the wound, you're going to fix it, right? How? Is it a miracle? No. Science doesn't have miracles. It's just mechanisms that you have to understand. And this is the way it's going to happen when it comes to the wound healing. Okay, so we will talk about bone marrow in great detail, but bone marrow, usually if you are having blood diseases and you're not going to be able to pinpoint it, the golden test that's going to give you an answer is going to be bone marrow biopsy or bone marrow aspiration. They're different. We will talk about the details what it means. In bone marrow aspiration, you're going to go in and stick a big needle in a specific area, usually the pelvic bones, the posterior iliac crest, and you're going to put the syringe inside of the bone, into the bone marrow cavity, and you're going to go in and get these precursor to white cells, red blood cells, platelets into the syringe, and you're going to be able to process it and look at those cells. You got bone marrow biopsy. In that case, you are going to have this kind of circular saw that's going to go inside of the bone, is going to get a piece of bone and all the architecture that is associated with it, and you're going to remove it. You're going to look at different things. Aspiration is going to tell you about type of cells that you have. For example, biopsy is going to tell you whether or not you have enough cells, or your cells have been replaced by fat. Is it hypercellular? Is it hypocellular? What's going to be the problem? We will talk great detail about it. Now, cytochemical stains, they are still being used. Time is changing. As I told you in the beginning, time is changing, so a lot of new techniques is coming. But these cytochemical stains were the way that people like me learned to identify leukemia. Because different group of leukemia are going to respond to different stains. So we would be able to go and use different things. For example, I have a leukemic patient. I look at the bone marrow. I don't know, is it leukemia, is going to be lymphocytic leukemia, which is lymphocytes are affected, or is going to be myelocytic leukemia, which neutrophils are affected. All I have to do is that I do myeloperoxidase stain, because only neutrophilic family, the myelocytic family, is going to be positive for this one. If my leukemia is from lymphocytes, I'm not going to get positive reaction. Okay, this is the way that we would be able to narrow down what the diagnosis is going to be. This was the old fashioned. Okay, it's still being used, but it's getting out of commission gradually. I think that by the time, within 5 years, these are the type of things that you're going to be able to see. Okay, so if you have the opportunity to train yourself in these kind of techniques, cytogenetics, flow cytometry, these are the things that are going to be the wave of the future. Flow cytometry, for example, you guys are going to do reticulum. What is the time right now? Okay, let's see. You're going to do reticulocyte count in the laboratory this semester. You're going to count about 1,000 cells. Okay? When you are doing flow cytometry, you can do the retic count with flow cytometry. You count 100,000 cells. You guys are going to spend 45 minutes to count 1,000 cells. Flow cytometry is going to count the 100,000 cells within a matter of 10 seconds. You see how it's going to happen? If you are going to have 200 retic counts, it would take you a lifetime to be able to finish it. Flow cytometry, they put them in the machine, it's going to go there. So these are the technologies coming and taking over, which is a wonderful thing. As long as you know how to interpret the results, it's going to be a wonderful thing. More importantly, is going to be molecular techniques, cytogenetics. Because of the fact that we are learning that when it comes to diseases, especially diseases of the blood, majority of the diseases, they're going to find their origin in the DNA. Something, either you got a mutation in the DNA, or you got a major change in the structure of the DNA, chromosomes and things like that, that account for the disease that the patient is suffering from. One example is going to be the famous one. About 95% of people who are having chronic myelogenous leukemia, 95% is a big number, people, are going to have Philadelphia chromosome. So you guys are familiar with this. Everybody does it? Is there anybody, this is the first time they have looked at chromosomes in this manner? So these are your, humans have 46 chromosomes, for those of you who don't know, and they're numbered in pairs. One from your father, one from your mother, number 1. And they're numbered based on their size. The longest one is going to be number 1, the shortest one is going to be number 22, and the last pair is going to be the sex chromosome. You have 2 X's, you are going to be a female, you got XY, one of the Y is going to be smaller, you're going to be a male, okay? In the Philadelphia chromosome, that's going to be very important. You're going to learn about it, you're going to know exact mutation, what has happened, which genes are involved. You're going to learn all of those things, but not today. If you notice, there is a piece of number 22 missing. Where is that piece? Is in here, number 9. During the cell division, something happened. That piece of number 22 got cut, and it didn't disappear or lost, but it came and attached to number 9. Now, everything is going to be handy and dandy as long as everything is okay. But what's going to happen here is that it's going to get cut in the middle of a gene in here. You got a gene in here, and all of a sudden the cut is going to happen in the middle of the gene, and it's going to come and sit up here in the middle of another gene. So it's going to completely ruin the structure of that gene, and it's going to cause what we call CML, for example. You see it in the other leukemia as well, but it's much more prominent in the CML case. Okay? This is going to be cytogenetics. Okay? When I was being trained about 25 years ago, we used to take a photograph with real camera. We would take a photograph of these things. We would print these pictures this big, and we would get the scissors and start cutting the chromosomes and lining up like this. Now everything done digital. You don't even look at the camera. You just do clicking, and you can do all these things without anything. I mean, you can do it so rapidly, so efficiently, so nicely, and prettier. It looks much better because now they use color staining in order to find that different part of the DNA missing or not missing. So they're going to look very, very colorful as well. Okay? So just telling you what we are going to go in and do, and I think that we are done at this junction here. We're going to start from the lecture 2 next time. Is there any questions so far? Again, next time I will give you a few minutes in between, but today I wanted to make sure I finish this one, so I'm going to let you go 15 minutes earlier. But if you have any questions, come and talk. Okay? Thank you very much for your attention.